morning, ladies and gentlemen. I'm going to be talking on the molecular ecology of invasive tamarisk in South Africa. Just a brief recap on what tamarisk is. Tamarisks are trees that there are tall trees, and sometimes there are shrubs, and there we've got a picture of one of the Tamarix, ramisissima, that I sampled in a free state, and then here this is a picture of Tamarix ramisissima in Cape Town. Then there we've got a Tamarix usneoides in the northern Cape. And then in South Africa, we've got these 5 species that have been reported, of which only one is indigenous. And Tamarix, it is an old world genus that has invaded so many parts of the world, including South Africa. And it's from the family Tamaricaceae, and it's got 55 species. In South Africa, the plant is being used as firewood and also as timber for construction, but mostly the pink flowering tamarisk is being used as an ornamental plant. And more importantly, the indigenous species is being used in the mines for phytoremediation, whereby it is uptaking the salt from mine dumps and excreting it as a gypsum. And the pink flowering tamarisk has escaped its point of introduction, and it has been invading our riparian zones and also our water channels. And the reason why we're trying to identify actually tamarisk is just to see actually what is the species which is invading our areas, and also because we're trying to identify the populations of the indigenous species that can be sampled for planting on the mines. And previously, I did some studies using the morphological level and also the ITS region just to see the different species that we've got. And this is a phylogenetic tree based on the ITS region. And then the ITS region clearly separated the exotic species from the indigenous ones. However, there were questions raising concerning the hybrid status which were occurring in there, and also this region gave us a lot of polymorphisms, confirming that hybridization is happening in our South African Tamarix. However, we didn't know actually which ones were hybridizing with what. So because of that, then we went on to do a population genetic study, and then hoping that this will solve questions concerning the taxonomic issues, and also to look at the population structure in the country, and also to investigate the country of origin for the invasive species, and also give us a better indication about hybridization and introgression. Introgression is meaning that the individual, the hybrids that are back crossing with either of the parents. And that takes me to the aim of my study, which was to characterize the tamarisk species in South Africa while investigating the hybridization and integration using molecular markers. And these are the 3 different markers that I looked at. I looked at the nuclear region, I looked at the plastid region, and also the genome-wide fingerprinting of tamarisk. The DNA material was sampled throughout South Africa, and as you can see, we tried to sample as much as we could to have a clear indication of the different populations that we've got in the country. And most of the ITS sequencing and the plastic sequencing was conducted at French University. But the FLP fingerprinting was done in the United States in one of the USDA laboratories. And Dr. John Gaskin is the person who developed the FLP markers. And then Ms. Kimberly Mann and myself were fingerprinting the Tamarix. And based on the ITS region, and I just want to draw your attention here that all the specimens that are marked in red are actually identities which were confirmed using the FLP markers. And this changed the complexity of this tree. As you can see now that all the chinensis, ramisissima, and hybrids are now grouping in the clade which is actually well supported, and leaving all the indigenous Tamarix right at the bottom in a different clade. And these individuals which were thought to be hybrid previously have all changed to be of pure lines of the indigenous Tamarix. And the FLP result, the FLP fingerprinting resulted in 196 polymorphic loci, which is quite a significant number, and then we're quite happy because the more polymorphic loci that you generate, the more information you're going to get through the FLP fingerprinting. And the FLP results were actually analyzed in different ways, and then this is a press-per-coordinate analysis. It is a distance-based analysis that we're trying to look at to see which ones are actually closely related to what. And as you can see there, all the chinensis are grouping in the same place, and I just want to mention that there we've got the South African and also the Asian chinensis grouping together in that clade. And here we've got all the ramosissima grouping there. And as well, we've got the South African and the Asian individuals that are grouping together. And they're quite distant from the indigenous species in South Africa. And we've got a whole lot of hybrids in between the two exotic species. Then I went on to do a population genetic study, and this was analyzed using a software called Structure. And what we're seeing here, first of all, when I ran the Structure software, we actually it resulted in the 3 clusters, and then this was expected because of the 3 different species that I'm looking at. And then this graph here, what is showing us is that each vertical bar is actually representing each individual, and the individuals are grouped according to species. And the percentages here on the x-axis represent the genetic makeup of each individual. And for every pure line cluster, you can see that we've got more than 90% of the genetic makeup. We've got chinensis, ramosissima, and usneoides. And there we've got all the admixed individuals. And these have got mixed genetic makeup and they are hybrids. And for every individual that had less than 90% of the genetic makeup belonging to any of these 3 clusters were actually classified as a hybrid. And based on the structure analysis, then we went on to quantify and see the species composition in South Africa. And as we can see here, that we've got actually more than 70% of the parental genotypes in the data set that I was looking at, and all the rest were hybrids. And this is quite a fairly high number simply because of the many indigenous samples which were thrown in the data set. But if we take out the indigenous specimens, then we've got also we've got different numbers now. And then so now we've got less than 20% of each of the parental types in our invasion, and most of the invasion actually in South Africa is of hybrid status. And the good news that we get out of this is that only 3.5% of the hybrid found in South Africa are hybridizing with our indigenous species, and all the rest are just hybrid between chinensis and ramisissima. And based on that, just want to conclude and say that the molecular markers that I looked at have given us that we've got only 3 species in South Africa and the hybrids. And then also the genetic differentiation between the exotic and the indigenous species has been given, whereby the exotic species are closely related to each other, but different from our indigenous species. And then the Tamarix invasion in South Africa actually is dominated by the hybrid individuals, contrary to what was thought previously that it was one of the parent species. And also we've found out that the parental types of the exotic species are matching the genotypes from their place of origin. And then just looking at what has been done elsewhere, Dr. John Gaskin has fingerprint Tamarix in the invasion in the United States, and he's found that the invasion there is mainly with chinensis, ramisissima, and their hybrids. And then the biocontrol has been taking place already in the United States, and the raptor beetles have been introduced to control the species. And also they've got Tamarix aphylla, which is exotic there, and it's actually very similar to our indigenous species here, but it hasn't been affected by any of the beetles which are controlling these guys over there. So that is just a piece of cake information that we get out of that. And this is Dr. Norton from Colorado State University, who's been looking at biocontrol of Tamarix in the United States. And we visited some of the sites where biocontrol is taking place, and as you can see, these are all dying Tamarix in the Colorado River. I just want to acknowledge these people, and thanks to Prof. Marcos for giving me the chance to have a scientific holiday in the United States.
Any questions? Okay. Sid, hit it.
Yeah, so in the natural ranges, are these species impacted? Because your study actually probably is very interesting. In natural ranges, maybe these species don't occur sympatrically and they don't hybridize, but in invasion situations, you're suddenly putting them together and they're hybridizing, and it might actually have huge implications for invasion biology.
Yeah. Like you're saying, in their native range, they actually far apart, and there hasn't been any evidence of hybridization. And as opposed to the place where they are invading, where hybridization is taking place, and this is one of the key issues that we think has been actually driving the invasion wherever they go. Because as in South Africa and in the US, it's just all the same situation.
Okay, and you haven't found any other examples? No, not from Tamarix, but from other, say, lineages?
Yeah, sure. Other population genetic studies have been conducted proving that hybridization is actually driving invasion of plant species.